Good afternoon. My name is Aleksa Petkovic, and today I'll present to you some work that my colleague Marko Šušić and myself conducted in Chaudhary lab at NYUAD regarding the effects of chronic social stress on gene expression in lateral habenula and suprachiasmatic nucleus. Our main motivation behind this work is that mood disorders are on the rise globally. The current approximations are that around 20% of all humans display either major depressive disorder or some of related mood disorders. Now, out of these 20%, around 50% do respond to current treatment, but the other 50% do not. In UAE, the prevalence rate has been estimated to be between 12.5% and 28.6% in 2019, but there are no definitive evidences regarding this. Now, there is a significant generational factor, as youth do exhibit much higher rates of depression and mood disorders and other neuropsychiatric disorders, so we believe that this number will increase. One of theories regarding the onset of mood disorders is circadian dysregulation. What are circadian rhythms? Circadian rhythms are daily oscillations of our bodily functions and processes that peak at a certain time and then return to baseline. This way, homeostasis is kept. Now, for example, our preliminary work and work by some other labs show that, for example, temperature, which is a direct measure of circadian oscillations, is blunted in depressed patients, whereas, for example, stress hormone and melatonin either exhibit phase advance or phase delay respectively. It has been shown that circadian process and sleep process are different, but they do exhibit effect on one another. Now, we believe that stress exhibits an effect directly on circadian process and sleep process, but also these two regulate mood. There might be some other third direct way that stress exerts its effects on mood, and this is the aim of research of our project. Two main regions of interest for us are suprachiasmatic nucleus, or SCN, and lateral habenula, or LHB. Suprachiasmatic nucleus is master clock in the brain, which controls all other peripheral clocks. All oscillations in the body are kept relative to oscillations that are produced in SCN. Lateral habenula, on the other hand, is the main mood regulation center in the brain. Previous studies in hamsters, for example, show that there is a direct projection from SCN to LHB. This projection hasn't been confirmed in mice, but it has been posited for a very long time, and we believe that this projection is the one that regulates the effects of circadian rhythmicity on mood regulation. The chief method that we use is highly reliable and valid paradigm for induction of depressive-like behavior in mice. Namely, it consists of exposing an experimental mouse, smaller mouse, to a larger, aggressive resident mouse. They will fight for 10 minutes and the resident will dominate the experimental mouse. Afterwards, we will separate them by plexiglass divider, which allows for sensory contact but not physical one. After 15 days of repetition, we will measure social avoidance in experimental mice using some novel, conspecific social target. We will measure the time that the mouse spends near a particular mesh with and without social target. We will take the ratio of these two times, and we will then, based on this, separate mice as either resilient, which will show higher time spent near where the social target is, and susceptible, which will spend most times in the corner of open field arena when the social target is present. Afterwards, we'll select these mice, we will perform RNA extraction, and then we will sequence the RNA we obtained. We'll match the reads, we'll align them to the reference genome, and then count them, generating a count matrix that will be used in subsequent steps. The results show that our paradigm was successful in producing both susceptible and resilient phenotypes, and the SI scores show clear differences in social avoidance between the two phenotypes. We also measured the distance traveled by all the mice in the open field arena without the social target present as a control measure, as locomotion can be confounded in these kinds of experiments. We show that resilient mice and susceptible mice, all the mice that went through social defeat paradigm, exhibit lower locomotion than controls. But this doesn't confound our findings because the differences between resilient and susceptible mice and controls even are not significant.
An overview of our differential expression analysis demonstrates that stress-induced transcriptional phenotypic differences depend majorly on the brain region investigated, the time of day when samples were collected, as well as the interaction between these two factors. Namely, in the LHB, we can see that changes occur both in a time-independent or global manner during the day and during the night, whereas in the SCN, we can see that transcriptional differences mainly occur during the night, whereas very few transcriptional changes occur between the phenotypes, either in a time-independent manner or during the day. In order to investigate more deeply the transcriptional changes between our phenotypes in the two different regions, we decided to plot volcano plots for the most interesting comparisons obtained from our overview analysis. The first comparison was the time-independent comparison in the lateral habenula. As we can see on the volcano plots, the most transcriptional differences occur between resilient and control and susceptible and control mice in the LHB in this comparison, whereas there's very few differentially expressed genes in the susceptible versus resilient comparison for this time-independent analysis. On the other hand, the diurnal differences in the LHB appear to be almost exclusive to susceptible versus control and susceptible versus resilient comparisons, whereas all controls appear to be very similar to resilient mice on a transcriptional level. In the nighttime LHB comparison, we can see a completely different story. Namely, the resilient and control mice appear to have the most differences in transcriptional differences in the LHB during this time, whereas the susceptible and controls and resilient and susceptible mice do not appear to have any major transcriptional differences. Finally, the nighttime comparison in the SCN displays that really the bulk of changes that occur in the SCN transcriptionally overall occur between resilient and control mice, and we can only see this difference appear in the nighttime comparison, whereas other phenotypic comparisons do not seem to have any major changes in this region. In our study, chronic social defeat procedure led to the differentiation between populations of mice that are stress resilient and stress susceptible. The stress-induced transcriptional phenotypic differences that were found depended both on the region of the brain sampled, the time of day of the sampling, as well as the interaction between these two. Namely, in the LHB, we see that time-independent differences show phenotypic transcriptional differences between both phenotypes and controls, whereas we can only see differences in susceptible mice during the day and only differences in resilient mice during the night. In addition, the two genes that were found in our analysis, SCN5A, which is an alpha subunit of a sodium voltage-gated sodium channel, and KCNV1, which is an inhibitory subunit of a voltage-gated potassium channel, might explain the excitability differences that were found in the LHB in susceptible mice in previous electrophysiological experiments. And finally, in the SCN, the transcriptional differences that occurred in resilient mice during the night only might explain the buffering active resilience mechanisms that were proposed by previous reviews. Namely, the SCN is a main circadian timekeeper, and in order to it needs in healthy mice to effectively buffer against stress perturbations on downstream regions. And overall, we believe that our results will help guide future work in dissecting the involvement of both the lateral habenula and the suprachiasmatic nucleus in circadian timekeeping depressive disorders, as well as their interactions. You can find our email addresses in the description of this video, so please do not hesitate to contact us if you have any questions. Thank you very much for your attention.